So you've heard from everyone on our panel today. We have Harrison Specht and Nikolai Slavov, two of our co-founders, and then Kevin McDonnell, who is one of our first team members to join PTI. So we'd like to open up the questions again, both in person and online. I'll grab my computer. But I would like to open up, of course, with some questions, and I'd like to say that these questions could be anything technical that you've seen during the talks that maybe has come to you since our speakers have, maybe since our coffee break. But also, this is the second birthday of PTI. So if you have any questions you're curious about Parallel Squared, then please feel free to ask. But I'll get it started with kind of a general question that I'll ask each of you to answer. Is that since you've all been present for the entirety of PTI, can you share what you view as one of the greatest achievements, maybe personally or as an organization?
Sure, I can start. I think that since starting PTI, I think one of the greatest realizations has been bringing the team together. We're very fortunate to be able to bring together people with a very diverse set of expertise to work on a problem like the ones you heard about today. So I'm incredibly thankful for the team that has decided to join us on this journey.
I had so many things to choose from that I have to confess being challenged by that question in a way that I rarely feel challenged. So thank you for the challenge, Sarah. But I'm going to pick on two things. One is I'm really quite pleased as to the ability to detect increasing number of copies for peptides labeled with this and that. I think it wasn't at all clear that this is going to be feasible to do by exploring relatively limited chemical space. And to me, that has been a very rewarding early observation from the team on the go. And the other thing that I'll choose to highlight is working with a team and that is not afraid to tackle bulk questions and to find that sometimes what I try isn't impossible. I think having this kind of attitude is very empowering, certainly something that I always enjoy myself, and being able to see this broadly shared visuality makes it an environment that I tremendously enjoy.
Yeah, just to go, I think the biggest thing would be is the team as well. I guess I have to be part of setting up the lab for this as new experience. And seeing how it has grown, or I guess since I joined, on the caliber people are joining Hyperlogics to run it parallel and accelerate. As you've seen here, we're going to get some results and why to show here by reading section to see as do you call it, we all agree. So.
Well, I that was a bit of a reflection. So moving into the next, several years, of course, and beyond, what are you most looking forward to at PTI?
You have the hot seat.
Yeah, no, I just can't think of an answer. I mean, the easiest, self-interested answer is I'm really curious about the tags. Tag 1.0, Tag 2.0. I think that, like Nikolai said, it's a really exciting discovery and gratifying to run a small assay on hundreds of compounds and find something that is working very well, and excited to expand on it. So technology-wise, that's my answer.
So what I would like to see happen is that we make proteomics much more scalable and extending far beyond the analysis of the abundance of protein products and more geared towards identifying other functional aspects of proteins, their modifications, localization within the cell, interaction with other proteins, and doing this not just through the analytical capability of detection and scalability, but really dovetailing this and driving those innovations with well-motivated biological problems. I think that being able to combine those two things together that are rarely coexisting in the same place is something that to me makes PKI very special. And I would like to see us doing this in a way that is compelling and helpful to others to adopt to their research. And that brings me to another part to this answer that I cannot resist adding, that beyond what I want to see happening at PKI, for the PKI is what's happening in the broader research community, not just what we do, but empowering others to start analyzing proteins directly so that we can increasingly replace indirect inferences with direct measurements, with data that are well positioned to start characterizing large populations of diverse patients so that we can detect and analyze comorbidities and have ultimately better understanding of disease mechanisms so that they can better predict outcomes.
Yeah, I guess from a computational point of view, I think we're generating now these large data sets, and the challenges that pose and all the new tools and maybe methods we need to develop, I think is exciting for me from a computational point of view. But then I guess we've heard about a lot of different technologies that we've been developing in parallel, to use the name. But now we get the opportunity to combine all these, so we can combine the different tags, we combine the different analysis, bite into these large single cell data sets, increase throughput, increase depth of coverage. And so combining all those together now is the next exciting chapter.
Any questions from the audience? Andrew's got one. What's up, Bill?
So at the outset of developing multiplex tags for DIA analysis, the question was always, how far can this be taken? How many single cells do you think you can run in parallel, at the same time, or, kind of per unit time? There's a limit to how short you can make gradients. Has, through your development of this first round of tags, do you feel like that an answer that has clarified a bit, or do you still feel like, it's a little bit more work to kind of see how far you can push the limit?
I think that's a deep and insightful question that has aspects for all of us. But let me take a quick crack at that. So, to some extent, I think there is an implied aspect of it, a tacit aspect, how far we can push the multiplexing without seeing downsides in terms of coverage and quality of quantification, which is very much part of our ambition, what we try to do. Of course, if we answer the question how far we can push it, if we're willing to take compromises, the answer might be different. Now, in terms of how far we could increase multiplexing, there's a side of making the molecules. How many barcodes can we make by using new codes, multiplexing by using partially overlapping isotopic envelopes? I think my estimate for that is at least 100 without going to very complicated molecules. But as Jason showed today, we may not need to make that many molecules if we combine it with alternative orthogonal dimensions for multiplexing, such as time. Perhaps it can be more economic to make a 20-30 plex and combine that with a 4 or 5 time plex, and that would give us a really good perspective and very good capability to parallelize. And of course, there's a challenge of counting the associated increase in number of ions and data complexity gives you without actually getting to answer the day's modeling aspect. But to add on the side of instrumentation, we have to worry about instruments having the capacity to count billions upon billions upon billions of ions. As I mentioned earlier this morning, the scale of the proteome is huge. And our friends at Bruker and Thermo Fisher come up with new and larger capacity, faster detectors every year. So I think at this point, capacity-wise, third commercial instruments can probably handle very well at least 30 plex for single cells, maybe 5 plex for bulk samples. But I see absolutely no physical limitations, practical limitations in the future to increase those capacities, and I am confident our friends are working on that.
Yeah, I guess to speak to the computational side of things, I showed, I think it was in my second or third slide, an ion map, and I guess as you multiplex, you're increasing the complexity of that ion map. Every single spectrum you take, you increase the number of peaks. And so, as you can see with droid modeling, it inherently can model those if they're the exact same fragment. But if you have a low resolution mass spectrometer and you have a high peak next to now a lowly abundant peak, and are you able to model, I guess, that interference between them? And someone asked about the Astrum high resolution earlier, and it's like, that can account for some of that, but as we exponentially or go to 100-plex, now we have 100 times more likely this interference happening. And so we don't know where the limit of that is. We haven't really we've seen that yet, but excited to see where it goes. But that may be a challenge for the future.
We certainly like challenges. It's very exciting to be on that forefront, pushing the limits and finding where they really are.
You have another question out here? Unless Harrison kick.
The only thing I'll add to what Kevin and Nikolai said is I think our theoretical expectations from the beginning, Andrew, were pretty similar to what they were 2 years ago, but I think we have a lot more clarity on exactly how we're going to test them. I think we got some wonderful collaborators who are helping us design, and we're learning a lot about how to design exactly the tests, what we want to do to get that answer.
I have 2 questions. The first question is regarding to you're studying Alzheimer's and looking into the disease mechanisms. How do you think, and this data is coming from brain tissue donated by patients, how do you think about testing it and your hypotheses in different disease models going forward? And then the second question, not related, is you're generating a lot of really rich data. How do you think about standardizing it for future machine learning models for diagnostics or things like that?
All right, I'm happy to take this one. I think the first one is about the challenge of testing mechanistic hypotheses from observational data. If we collect postmortem tissue from human patients, how do we test a specific mechanistic hypothesis? Perhaps the most readily available avenue for us is by using iPS-derived neurals, where we can do experiments. And we can also test it in animal models through our collaboration with Brent Hyman and other colleagues. Fundamentally, I think we have to acknowledge that when it comes to neurodegenerative diseases, there isn't a great modern system because iPS neurals are not human brains. They're useful for testing things, but they're neurals in a ditch. They're not brains. Mice are not humans. And one of the very pervasive growing awareness in the community of neurodegenerative researchers is the animal models, in some cases even primates, have major limitations in terms of recapitulating human disease. So we'll use the best methods available to the community, to anyone, primarily also that they mentioned, but acknowledging that that they do have limitations. And with regard to data standardization, we are very keenly aware of the importance of using strong benchmarks, establishing community standards. We recently led the first effort to develop best practices and recommendations for single-cell proteomics, working with other leaders in the community. And when we feel that there's a need to do that in the contexts of diseases or different data types, we'll certainly initiate these community-driven initiatives to establish standards. But we are very mindful that we're using resources. We're fortunate to have access to these resources, and we should make the most of them, which includes making the data that we generate and collect available for others to analyze and interpret. That's a very important part of our mission.
Yeah, I just add to that in terms of a machine learning context. Nobody from a machine learning background wants to look at a load of raw spectra and say, okay, how can I train my models? So I think having interpretable data, not just giving them the raw data, but actually something someone could use is super important and was something we need to do.
I think the final aspect we can add to that is the analyses specifically available to single cell proteomic studies are well applicable to brain studies like Mammoth described on single neurons, single microglia. We're excited to apply those sort of analyses to the data we were able to collect with our collaborators.
Hello. I have a question. I heard you mention in future directions localization. I'm just wondering if you have an interest in adding imaging mass spectrometry or deep visual proteomics to your workflow in the future.
Yeah, these are interesting possibilities. I think in terms of localization, what I think more about is being able to either label with different mass tags proteins in different parts of the cell, because this works very well with our ecosystem. We can synergistically enable as part of developing other methods. To the extent that we can physically isolate different cellular compartments at high throughput with other methods, such as laser capture microdissection. That's certainly a possibility, even though those approaches have generally been difficult to adopt at scale in many people's hands. But mass spectrometry imaging, of course, is a wonderful method for analyzing lipids, metabolites, with proteins that is more limited and harder potentially with peptides. But I think our foray in this direction and choice of method is going to depend on the specific biological problems that are motivating it, and what will be most effective and informative for the ground.
We were a couple questions online, and not get back to the audience. One of them is a lot of clinical or translational labs use affinity-based proteomic techniques because of ease of use. How can PTI's techniques be made as accessible as possible for less technically experienced labs? And I think we talk a lot about community. So broadly, how can we get the community more involved? How do we make this accessible?
That is an excellent question. Would you like to answer, Harrison, or should I? Would both. All right. So let me take the first cut at that. So population proteomics, certainly in the context of plasma, is something that has been of great interest for decades and has received renewed attention and interest more recently, especially in the context of affinity reagents being developed for a larger number of proteins and being scaled to analyze samples from tens of thousands of patients. Mass spectrometry has the capacity to be much more specific in detecting exact amino acid sequences and providing more information about different proteoforms and different protein products. It certainly has struggled to scale to the analysis of tens, hundreds of thousands of samples, and this is exactly a place where our multiplexing technologies, our approaches to plotting, they can parallel, can make a huge difference. They can make the difference between something being technically possible but economically unaffordable to something that is able to not only increase data quality but also reduce price. And I think there was a very important element to that question Darius chose as to not only the feasibility and the price, but also the barrier to implementation. And when it comes to mass spectrometry, it's a very powerful, very sophisticated technique that requires expertise to understand it used correctly. And we can do two things in that regard. One is to educate the broader community. We have been very passionate for that for years, certainly in the context of single proteomics more generally in mass spectrometry. I think there's a lot to gain by explaining what are the capabilities and limitations and allowing more people to have access into use the technique. And the other one, of course, is having more state-of-the-art facilities such that can help with this analysis, whether this is CROs or whether this is core facilities or other organizations. PTI is not a CRO or core facility, but we are certainly very happy to help to partner and to enable others who use own technology to enable CROs and core facilities to do this analysis. And they think the combination of those different things, education plus facilities that are able to perform the analysis in make the technologies more accessible. And the final thing that I'll say in this, like you answer, sorry, Bernal, but it's a bit complex question, is that if we think of what happened to a text generation sequencing and that being so broadly accessible and available, is that there was a sustained period of decades of major investments from federal and other sources that eventually resulted in very standardized and robust technology. And if one wants to get to a point of having robust and standardized technology, having this community engagement and long-term sustainable support is very helpful.
I'll add on to what Nikolai said, and that I hope that you're in the position to offer a lot of that. I think that not only are we trying to put out technology and open up platforms for collaboration, but given the position we are in as a nonprofit for forming collaborations, I think we're also in the position to well sustain them, to do the sort of education that Nikolai was talking about, not just writing papers, to get an idea out there, but writing papers, get the idea out there, and then support the people who would adopt the idea, distribute it, use it. So I think that hopefully the model of this organization will prove to be effective towards those ends as well.
I may just add slightly, I'm not sure if this was in the question at all, but from a computational point of view, I think with the tools we're developing here, from early on, we had this motivation. We wanted to make things open source. I think it's super important that we could do that so people can see and understand, what it is our models are doing and how they work and what is generating data. And I was very lucky. My PhD from other groups had open source material that I could use and utilize and build upon. And so having that available to let people, come into the field, see what actually an algorithm is, how it works. And then alongside that, the onus is on us now to also make it usable enough so that if you don't need the technical details to be able to run the source code, you need to be just something that's really accessible, really usable. And so the onus is on us now to provide that platform for people.
So at the beginning of the conference, you spoke about the space of billions of proteins to be analyzed, and some proportion of the billions of proteins have not yet been identified. And I'm wondering if you folks can kind of speak to how you're planning to identify new proteins. I see, peptide fragments, or is this happening? Has it happened? Where do you see this happening in the future?
It has happened. This is a research project that actually took place in my lab here at Northeastern, and something that I'm very excited about. So it has to do with how do we know the sequences of the proteins. When we say that there's a human protein, we have database with a sequence. What is the origin of that database? The origin of that is a sequence of DNA usually, or potentially sometimes RNA, more rarely, but then by using a genetic code has been translated into a mono-expected sequence for the protein. And how do we know the genetic code? This is something that in the 1960s was worked out using in vitro experiments of synthetic RNAs translated by E. coli cell extracts. So this is not translation happening in vivo in mammalian cells. The E. coli translation apparatus, tRNAs, ribosomes, are very different from ours. There are 10 times fewer transfer RNAs, and the ribosomes are different. So as to how are the proteins translated in the human body, it has been very difficult to do these measurements directly because identifying protein sequences is so difficult. Most of the time we don't de novo sequence proteins. We predict what the possible sequence is, and then we match that to existing spectra, and based on that we can tell, did we detect it, did we not detect it? And one of the reasons why this is so difficult is because our spectra have not had enough fragments to do the de novo sequence identification. To the extent that we're able to make tags and molecules with enhanced fragmentation patterns, we are in a better position to do de novo sequence identification. So the data that we are generating, including the technologies that we are developing, are going to make direct protein sequencing much more powerful, and you'll start learning more and more about the full scope of sequences of human proteins. And we have additional clever ideas that we are very excited about that can further empower the ability to de novo sequence proteins without needing to have an existing hypothesis for all the sequences.
I think I can close this out with one more question from online. The presentations described exciting methodological improvements in mass spec. Do you anticipate innovations or breakthroughs in MS instrumentation over the next 5 to 10 years? And I think we can preface that, being that we are not the instrument vendors. We can't anticipate, or, we don't know what to expect from our vendors entirely. But as Nikolai already hinted to, capacity is one route of, possible road of improvement, especially as we increase this multiplexing. But I'll turn it over. I'm going to make you start, Harrison, and if you have anything else you want to expand on.
Thank you. Can this become a wish list?
Making notes.
Yeah, thank you. I think there's a lot of exciting possibilities. You saw our application of some of the mass spec ancillary technologies like iMobility in Maddie and Mark's presentation. I think technologies like those are very exciting in the sense that they offer more dimensions for analysis. We had to have a theme of the day and think about scaling proteomics. It's all these other dimensions for multiplexing that may be accessible. And Jason well demonstrated multiplexing in the time domain. So I think that's an exciting possibility for people putting together these LCMS systems.
Absolutely. I very much expect improvements in instrument hardware. So this is something that instrument vendors and very creative scientists, colleagues are constantly working on, challenging the capabilities. And I'm also very pleased to have seen a lot of enthusiasm in these colleagues and appreciation for the technologies that we're developing. They realize that this holds tremendous potential for increasing the capacity, the power of proteomics, which can open new applications, new markets. So this is something that is very synergistic and beneficial for companies as well as driving wider adoption, more scalable adoption of their instruments. Because if an instrument produces 10 times more data and has the same price tag, there are more customers for that instrument. And from that perspective, we work closely and discuss with physicists and engineers and corporate leaders at Bruker, at Thermo, what are some of the improvements that we think can really empower multiplexing and other applications. We can certainly benefit from the ability to handle more ions. As we multiplex more and more samples together, we need to be able to handle more ions. Gains in resolution are always welcome because they help us mitigate interference, which can become an increasing problem as we increase ion density. Certainly improvements in efficiency of transferring ions, converting the electron to detectors, can give us very substantial gains in sensitivity. And that is important for single-cell proteomics, for single organelle proteomics, for PKM analysis, being able to more efficiently capture fragments with different M over Zs. It works very synergistically with improving fragment generation so that we can ultimately detect more fragments and more comfortably identify peptide sequences. So I think there is a large scope for improvement on the side of instrumentation, and many of those improvements are quite practical, feasible, and likely to happen within the next years and help us and help the rest of the community. Some of those improvements are going to take longer to be realized, but I'm certainly counting on collaborations with our friends who made the instrumentations and that is required to achieve our mission to make proteomics much more accessible and much more powerful so that we can drive discoveries and help understand disease.
Yeah, I can give the short answer, more resolution. Just agree with what's been said here, more capacity, and definitely that one, sampling more ions. I think particularly for this modeling approach we talk about, having that expectation is so important. But if you're sampling very few ions, suddenly you get into sampling statistics and you don't, it may be random or more variable what your actually fragmentation spectra you see. So if we could get greater number of ions into the mass spectrometer, you have a better expectation of fragmentation variety, getting more accurate identifications.
All right. Let's thank our panel.